This is Murat Kuscu. I'm an assistant professor at the Department of Electrical and Electronics Engineering at Koç University. So I would like to first thank Dr. Yan Shadeng for organizing this wonderful event and inviting me as well. I would like to thank also all the speakers for their insightful and engaging talks. I really learned a lot, so thank you. So my contribution will be an attempt at answering the question of how we can get practical with molecular communications by engineering microfluidics, nanomaterials, and ligand-receptor binding interactions. So at Koç University, I'm leading this CALICO Lab, Nanobiophysical Information and Communications Laboratory, where we perform theoretical and experimental research at the intersection of information and communication technologies, nanotechnology, and biotechnology. This picture roughly summarizes what we try to achieve. This is actually Internet of Bio-Nanothings. So we want to create seamless communication interfaces between heterogeneous bio-nanothings, like natural biological systems, like synthetic or engineered biological cells, or abiotic devices like nanobiosensors. So we are engineering interdisciplinary solutions to interconnect non-conventional things with each other via non-conventional methods like molecular communications in non-conventional environments like inside human body, and for non-conventional applications like continuous intrabody health monitoring. So we use nanotechnology to build microfluidic prototype molecular communication testbeds and develop practical transmitter and receiver architectures for molecular communications. We draw on biophysics to understand, model, and engineer molecular interactions at nanoscale, like ligand-receptor binding interactions, and optimize them from a communication perspective. And ICT is our main framework where we want to connect this nano and bio and give birth to IOB-NT applications. So I'm not going to go over all the IOB-NT applications here, as I assume that the audience is well aware of these practical applications, potential applications, let's say. For example, the smart drug delivery or the continuous health monitoring. These are the applications that attracted the most attention in the literature. But the point is that we are not yet there, right? So there is no any practical deployment of any IOB-NT system to date. But it might be useful to have a classification of what we call bio-nanothings. What tools do we have for IOB-NT? And I like to divide them into three main categories. The first category is passive molecular agents. So these are primitive devices consisting of single or a few molecules like fluorescent nanoparticles, fluorescent dyes, etc. The point is that they are not able to perform active communication or any other function actually, but they require an external stimuli, energy source. And once stimulated, they randomly start to exchange some sort of energy between each other. And the second class is abiotic devices, which are mostly based on nanomaterials like graphene. And of course, these are much more complex than the first group. They can perform active communication. They may have memory. They can perform computing, etc. And the third group is biosynthetic devices, which can be synthetic or artificial cells, like engineered bacteria. The point is that they are inherently capable of communication by molecular means. So my research has addressed all these three groups of devices to varying extents. But in this talk, I will focus on abiotic devices and the biosynthetic devices that can actually perform molecular communications. So what is molecular communications? Is the transfer of information via the exchange of molecules, right? Just like the living cells communicate. So we are dealing with a radically different method compared to conventional electromagnetic communications here. So we need to approach this new modality with a multidisciplinary research toolset. And actually, we did a lot from theoretical aspects, such that we developed channel models, we developed detection techniques, we developed simulation frameworks, we derive the theoretical performance limits. But in doing so, we relied on and we are actually relying on still many simplifying assumptions, right? So once we start to actually build practical molecular communication testbeds and start running experiments on them, we've seen that even in the simplest settings, our theoretical predictions do not fit very well into the empirical results. So we need to go back, revisit the theory, and refine it. Also, some of these experiments revealed some interesting physical phenomena that can be exploited to diversify our molecular communication techniques. So these 2 points actually highlight the importance of having practical testbeds for molecular communications. So the journey of building testbeds has actually started like 10 years ago with this tabletop molecular communication testbed developed by Professor Andrew Ackworth's group. And later on, many different research groups have taken different approaches to build testbeds of varying sizes. Some of them are microscale, but most of them are microscale, big testbeds. Some use gaseous medium as the communication channel, like air, and some prefer liquidic channels, aqueous channels. And as the information molecules, some rely on alcohol molecules, but the others use, for example, hydrogen ions or magnetic nanoparticles, and even the fluorescent dyes. And not all of them are actually based on abiotic components, but a few of them actually use biosynthetic devices like engineered bacteria as a transmitter or receiver. Okay, these testbeds have been really useful for refining the theory, for developing more accurate channel models, more practical transmitter-receiver architectures. But of course, there's still room for improvement. For example, if we have more control over the design of these individual components like transmitter, receiver, and channel, we can actually optimize their architectures from communication perspective. And with that, we can develop custom-designed components to diversify the MC applications or MC scenarios that we can test with these testbeds. And extending the spatial temporal resolution is also important, especially for the transmission and propagation for molecular communication systems. And also the scalability to micro nanoscale is another challenge for transmitter and receiver architectures. Is not a great deal of challenge actually for channels because we already have microfluidic channels, right? The other point is that if we look at all the practical testbeds developed so far, we can see that the focus is particularly on the propagation of the molecular signals, maybe except for the biosynthetic testbeds. But with that, we might be actually missing some important molecular interactions like ligand-receptor binding interactions, which can be a challenge and also an opportunity at the same time for molecular communications and Internet of Bionanothings. So as an alternative, we developed these receiver-centric microfluidic molecular communication testbeds, which is not able to solve all these problems, but I believe it takes an important step forward to address these challenges. So this is the diagram and the picture of our testbed. We use a commercially available microfluidic pressure controller as the transmitter and a microfluidic chip that contains both the molecular communication channel and the receiver at the bottom, which is actually a field effect transistor-based biosensor, actually a DNA sensor. So we use DNA molecules as the information molecules in this case. And for the receiver, we built a field effect transistor-based biosensor, also known as BioFET, which can convert the concentration of chemical molecules to electrical signals. So in BioFET, the binding of these ligands, these yellow dots, to the surface receptors modulate the channel conductance, the conductance of the transducer channel via field effect. So the output current of the receiver becomes a function of the ligand concentration. So we prefer using graphene as the sensing material in this transducer channel because it's highly sensitive. Its biochemical sensitivity is very high. It is flexible and also it is two-dimensional. So it brings integration advantages. So we first fabricate the field effect transistor based on graphene on a silicon oxide substrate. We use a monolayer graphene produced through a chemical vapor deposition technique. And then we built the PDMS-based microfluidic channel and integrated into the fabricated graphene transistor. After that, we performed the biofunctionalization of the transistor with receptor DNAs or the probe DNAs, which can complementarily bind target DNA molecules, single-stranded DNA molecules, which encodes the information. So these receptors give the receiver a very high level of selectivity for the information molecules. And then we determine the sensitivity and the selectivity of our receiver, and then perform some communication experiments by encoding the information into the concentration of DNA molecules following an on-off keying approach. And at the receiver, we observe a substantial level of intersymbol interference, limiting the communication rate substantially, such that we are talking about hundreds of seconds just to send one bit of information. Okay, maybe we don't need very high data rates as you have in 5G networks, but we should definitely do something about that to extend the application range of molecular communications. So moving forward, what are the problems with our testbed? First, we have a problem with the transmitter. So in these experiments, we use a microfluidic pressure controller to modulate the injection rate of information molecules and the buffer into the channel. But we actually don't know what gets into these microfluidic channels, how we shape the concentration pulses through this Y-shaped channel entrance. So if we don't know that, how we shape the channel, how we shape the concentration pulses, we cannot actually test the performance of modulation detection techniques accurately and develop new ones. So we turn our attention to practical microfluidic pulse shaping techniques, and the hydrodynamic techniques were our first targets. So this technique used only the interplay between different hydrodynamic forces supplied through different inlets of the channel to shape the chemical waveforms inside microfluidic channels. So we are, for example, working on this hydrodynamic gating technique, where the information molecules supplied through this inlet continuously are switched on and off through this buffer flow supplied through this gating inlet. And we show that through both simulations and experiments, it is actually able to generate very short and high bandwidth concentration waveforms inside microfluidic channels. What about the receiver, the main component of our transmitter? So if you look at the receiver's response to concentration pulses of varying durations, we see that it takes significant amount of time for the receiver response to return to its baseline current. And this is not expected in a microfluidic convection diffusion channel because all the molecules remaining in the channel are very quickly purged from the channel. So this implies that the problem might be associated with the receiver. And in fact, it is caused by the low dissociation rate of the DNA molecules. So they stay in the hybridized state for a very long time. So it takes a long time for the receiver to return to its baseline. So we turn our attention to ligand-receptor binding interactions to understand their effect on the molecular communication performance. And by the way, these ligand-receptor interactions are not only important for our testbed or for the biosensor-based molecular communication receivers, but also they are important for biosynthetic devices because ligand-receptor interactions are widely utilized by biological cells as an interface with the channel for sensing and communication. And in the drug design and biosensing context, we want these ligand-receptor interactions to be very strong, such that we can increase the sensitivity of our biosensors, and also we can actually target the disease cells more effectively. But this is not the case in molecular communications because we are dealing with time-varying dynamic signals. So if they bind very strongly with low kinetic reaction rates and high affinity, they will stay bound for a very long time. So we will not be able to sense new incoming signals in molecular communication context. And this will ultimately lower the bandwidth, the channel bandwidth, which is already limited by the low pass characteristics of the diffusion-based molecular communication channel. And bandwidth is not the only performance metric that is affected by the interaction parameters, but our studies show that the sensitivity, the dynamic range, selectivity, and the noise characteristics of the receiver are also strongly correlated with the interaction parameters, like their affinity or kinetic rates. So all these trade-offs actually point to a multi-objective optimization problem regarding these interactions. So these ligand-receptor interactions not only bring a challenge, but also they provide some opportunities for designing new molecular communication techniques. So we are living in the age of de novo protein design, synthetic receptors, and aptamers, which means we have the tools to tune the interaction parameters, tune the kinetic rates, tune the affinity. So in a sense, it actually provides a new way to develop more practical molecular communication techniques like modulation detection techniques or adaptive molecular communication networks. And also it gives us an opportunity to address multi-access and coexistence problems in crowded molecular communication networks. So in the next couple of slides, I will try to convince you that these ligand-receptor binding interactions can be actually engineered or exploited to enable new molecular communication techniques. Now we are working on this frequency domain detection technique, which actually exploits the fact that different reaction rates of different types of molecules with the same type of receptors are actually reflected to the power spectral density of binding fluctuations or binding noise as different cutoff frequencies, which can be observed at the electrical output of our receiver. So we developed this frequency domain detection technique and compared its performance to a time domain detection method under the interference of a similar type of molecule, which can actually interfere with the receptors. And we show that it brings lots of advantages, especially when this interference drives the receiver into the saturation region. In the case of biosynthetic devices, we have access to the individual receptor states, like their bound and unbound states, actually the continuous history of these binding states. So these receptors fluctuate between bound and unbound states, depending on the ligand concentration. It's actually a random process. And we show that the bound and unbound time durations of these receptors actually provide much richer information compared to the instantaneous number of receptors or bound receptors, let's say. For example, the bound time durations are informative of the concentration ratio of different types of ligands coexisting in the channel, and the unbound time durations inform about the total ligand concentration. And by combining these two statistics, we develop a channel sensing method, which can sense the concentration of different types of molecules at the same time. And it is very important for molecular communications because all those biochemical environments are actually full of many different types of molecules that can interfere with the molecular communications or that can bind the same receptors with different reaction rates. But with this technique, the receiver can discriminate between different types of ligands and can actually eliminate the interference, molecular interference. And with that, actually, we can enable multiplex molecular communications as well, or we can develop new multi-access communication techniques for molecular communications. For each of these techniques, we are also developing synthetic receptor designs and also chemical reaction networks that can sample the bound and unbound time durations of receptors and also perform the arithmetic operations that are required for decoding these messages. So recently we focused on this ratio-based modulation technique, which was actually proposed by another group 10 years ago. But since then, nobody actually studied that. I don't know why, but it's actually a very simple technique. The information is encoded into the concentration ratio of two different types of ligands. It is simple, but it can be quite robust against dynamic channel variations or dynamic variations in the transmit power, as long as these variations affect both types of ligands at the same rate. So their concentration ratio remains the same, whereas the individual concentrations might change. So in that sense, this ratio shift keying modulation can be quite advantageous compared to the concentration shift keying. And what we did is to actually show that by only sampling the bound time durations of receptors with a very simple technique, we can actually very accurately decode the information encoded into the concentration ratio. And we calculated the end-to-end channel capacity with this RSK modulation and compared it to the CSK modulation when the transmit power of the transmitter is limited. And lastly, these ligand-receptor interactions can be exploited to enable adaptive molecular communication receivers. So in nature, there are various biological mechanisms like this allostery that can dynamically and adaptively tune the interaction parameters, like their kinetic race or the receptor curve, let's say. For example, by tuning the dissociation constant of receptors for a certain type of ligands, we can actually dynamically tune the dynamic range and the sensitivity region of our receiver for the information molecules. And this is very important for time-varying dynamic molecular communication channels. And we actually simulated such cases when the receiver has these adaptive receptors in the presence of, for example, background interference or degrading enzymes or intersymbol interference. And we show that the adaptivity brings significant benefits for molecular communications. And lastly, to conclude, it is obvious that we need multidisciplinary collaborations to get practical with molecular communications, because the problems we are dealing with cannot be only addressed by communication engineering tools, but we also need the tools and concepts from nanotechnology, for example, biotechnology, and even the philosophy of sciences to be able to answer questions like what is biological communications, what purposes does it serve, or how to distinguish between a mundane molecular interaction and a purposeful biological communication, etc. So we need to unleash interdisciplinary synergies between these fields. Actually, as a humble step to this end, we published a perspective article recently with researchers from nanotechnology, biotechnology, and philosophy of sciences. And we try to identify the challenges for creating such synergies. And we also propose a potential pathway to overcome these challenges. And in a similar direction, we just announced a special issue in IEEE WMBMC. So I would like to invite all of you to submit an article to this highly interdisciplinary special issue. Thank you very much.